Now we are going to study the reactions of amino acids by learning about the formation of a Zwitter ion. By definition, a Zwitter ion is a dipolar ion that carries both a positive and a negative charge as a result of an internal acid-base reaction that occurs in an amino acid molecule. As we can observe, if an amino acid is not ionized, we can clearly observe its carboxylic acid and its amine group or amino group. Now, when a Zwitter ion forms due to changes in pH, what we can observe is that the amino side gets protonated and it gets an overall formal charge of +1 at the nitrogen. And then the carboxylic acid will lose a proton. And then the oxygen that used to have the hydrogen, in other words, the proton, is going to become -1 formal charge. Since a Zwitter ion specifically has a +1 and a -1 formal charge at those atoms, when you add those formal charges, that equals 0. So the net charge for a Zwitter ion is neutral. Under different pH conditions, a Zwitter ion can act as an acid or it can act as a base. Let's look at two examples where there's variations in pH, and we will observe how the Zwitter ion will interact with these different conditions. Let's say that, for example, we have a solution in which the pH is now acidic. Under acidic conditions, in the solution, we will have the formation of the hydronium ion. The hydronium ion is specifically going to be acting as an acid. That means that since this is going to be an acid-base interaction or reaction between the two molecules, then somewhere in the Zwitter ion, one of the atoms have to act as a base. Since the Zwitter ion acts as a base, specifically the oxygen atom at the Zwitter ion will be acting as a base, and we can observe overall that one of the hydrogens from the hydronium actually is now paired with the original oxygen atom in the Zwitter ion. Since the hydronium, meaning H3O plus, loses a proton, then what we observe in the product sides is that its conjugate base is going to be the formation of water. When that oxygen atom in the Zwitter ion receives that proton and now is linked to it, it makes this part of the molecule now have a formal charge at the oxygen atom of 0. So if now we look at the overall charge of what used to be a Zwitter ion that reacted under acidic conditions, we see that as a result, the product is going to have a positive net charge. What will happen if instead of acidic, we are under basic conditions? Let's say that now we have a pH that has increased, and now you have OH, meaning hydroxide, in your solution. When you have a base in the form of hydroxide in your solution, then the Zwitter ion will be acting as an acid. Specifically, the area of the Zwitter ion that will be donating a proton is going to be one of the hydrogens of the originally protonated amino side. When that happens, one of the hydrogens from here combines with the OH, and that's how we form water. Because nitrogen lost an H+, it goes from formal charge of +1 to 0. And now, when we look at the resulting overall charge for a particular amino acid that has reacted with base, we see that the product is a molecule that has a negative net charge. We can observe an application of the isoelectric point by looking specifically at human albumin. In figure 12.4, we actually have an image of blood samples. Human albumin is a major component of plasma, which is one of the things that we have in blood. Albumin has an isoelectric point of around 5, which happens to be a isoelectric point that is lower than the pH of blood, which tends to be 7.4. Since albumin has an isoelectric point around 5, this lower isoelectric point actually prevents the protein from becoming neutral and aggregating unnecessarily in the blood, meaning that since it's not going to aggregate, it's not going to precipitate out of blood. Let's continue talking about the reactions of amino acids. Now, let's discuss specifically peptide formation. Individual amino acids bond together, much like we observed previously when discussing organic chemistry, by the combination of a carboxylic acid and an amine forming an amide. So, if we go back into our knowledge of organic chemistry, we previously observed that if you have a carboxylic acid and you have an amine, and you heat them, you can form an amide linkage right here. And the linkage specifically is that covalent bond between the C double bond O and the nitrogen. When it comes to amino acids forming this amide bond, and you may recall that this reaction is called amidation. Now, this is actually called a peptide linkage, or in other words, a peptide bond. So when we have individual amino acids, and I'm going to zoom in into the reaction of two amino acids, we can see specifically that, let's say that we are going to combine a glycine amino acid with alanine amino acid. Overall, what we observe in this amidation reaction is that, let me highlight the items to make it more obvious. The oxygen atom from the first amino acid is going to go away and is going to combine with, let me just rewrite the hydrogens here. So let's just put it as NH2, and then let's put the third original hydrogen as an extended bond. So the oxygen atom in the glycine will combine with two of the hydrogen atoms from the next amino acid, and the combination of O minus and two H's is going to make water. Then our nitrogen atom that has a hydrogen is going to be covalently bound to the C double bond O of the first amino acid. So if I highlight those atoms, what we see here is that this forms a bond with this area, and that's where we have the peptide linkage or peptide bond. In other words, the amide bond formation between two amino acids. When it comes to amino acids, understand that the amino acid that is first written right after the first amino group, that is called the N-terminal residue within a peptide or protein. When it comes to what is the C-terminal residue, the C-terminal residue is going to be the last amino acid that is bonded to the right end of the molecule that it is connected. Let me do it once the peptide is formed. So the N terminus is going to be overall the amino acid that it is the first one in within a peptide. When it comes to the C terminus amino acid, it's going to be the last one that you can see that it is highlighted in pink. Know that overall, this is a very short peptide. So in the next example, when we actually draw examples that have more, I will again highlight what is the N terminus residue versus the C terminus residue. But for now, understand that the N terminus residue is going to be overall the amino, the alpha carbon with the R group, and the C double bond O that we have at the beginning of our peptide. And then the C terminus residue or amino acid is going to be overall the O minus C double bond O alpha carbon with its tight chain and the NH group within a peptide. Let's actually practice if we are given a particular sequence of amino acids, how to draw the peptides. I'm going to illustrate two different techniques on how to do this. In the first problem that we have here, we need to draw the peptide that has the amino acids proline, alanine, and lysine. Understand that in exams, I actually give you a version of table 12.1. Please make sure that you go over the in-class worksheet where I illustrate exactly the table that it is provided in exam. Pretty much the table contains the structure, the skeletal structure for the amino acid, the full name for the amino acid, the three-letter code for the amino acid, and additionally, it has the one-letter code for the amino acid. So make sure that you practice extensively because this table is going to be utilized in this chapter in many instances. So now that I know that I have the amino acids pro, ala, and lys, going to the table that is specifically in the in-class worksheet, you can actually draw those individual amino acids out. So right now, I'm actually going to draw proline, alanine, and lysine individually. Let's start with proline. When it comes to proline, and looking at the table that it is in the in-class worksheet, it's going to be drawn as NH2 connected to an alpha carbon, connected to a C double bond O, OH. And then from that NH2, what we have is a cyclic system that looks like five atoms overall. That nitrogen is positively charged. Once I have done that, then understand that I'm going to just erase the hydrogen on the oxygen in the carboxylic acid side. So this I'm just going to erase and then just put a negative charge. Let's write the next one, alanine. When it comes to alanine, alanine is going to be drawn as NH3, which I'm going to illustrate as NH2H, to be honest with you. And I'm going to put a positive charge. And then I'm going to have the alpha carbon with a methyl group, C double bond OH. But just like I did for proline, I'm going to erase this hydrogen and I'm going to put it as a negative charge. Next, I'm going to write lysine. When it comes to lysine, I have an NH3. I have the alpha carbon, C double bond O. I'm not going to write the OH. I'm just simply going to write it instead of OH like the table, write it as O minus, which is perfectly acceptable. And then for lysine, I observe that I have 1, 2, 3, 4 carbons. 1, 2, 3, 4 carbons. And out of that, I have an NH3. And that NH3 is positively charged. Now that I have written my amino acids, let me just do one more change. This NH3, let me write it as NH, and that's connected to H2. Let me just erase that bond. Okay. Now, how do I draw the peptide? As I illustrated in the previous example, in essence, what we have is that this oxygen connects to these two hydrogens, and when they come together, they form a water. Next, we see that this oxygen combines with these two hydrogens, and we form another water molecule. So instead of writing two individual water molecules, understand that you can write it as two H2Os. Now, in essence, to draw this peptide, all we have to do is now write amides from this C double bond connected to this NH. We're going to write another amide where this C double bond O is connected to this NH. Understand that once we make that peptide bond or that peptide linkage, the nitrogen no longer is going to have that positive charge. So when I write overall my final answer, I'm going to draw NH2, positively charged, alpha carbon C double bond O, and then I'm going to connect that to NH. So I keep it consistent. That is what I highlighted in purple. Then after that NH, I see that I have a CH with a methyl. So I draw a CH with a methyl. Next, I draw a C double bond O. C double bond. Let me not write the carbon, but it is implied that it lives there. C double bond O connected to NH. That basically is going to be this atom now covalently bond to this NH. And then I have an alpha carbon, and I have 1, 2, 3, 4 carbons connected to NH3, positively charged. And then, since I already drew this here, the last thing that I need to draw is that C double bond O, O minus. C double bond O, O minus. So that is the peptide that comes from combining a proline, an alanine, and a lysine. Overall, to be honest with you, once we have this given to us, these lines tell you how many amide bonds you need to write in. If we actually look closely at our final answer, we can see that there's going to be a pattern. And that pattern is nitrogen, alpha carbon, amide, alpha carbon, amide, alpha carbon, and since this is the last amino acid present, then it ends in a C double bond O, O minus. So what that means is that if I want to do the shorthand, so the faster way, instead of sitting down, writing individually the amino acids, then drawing the formation of water, and then drawing the peptide, we can actually write it in a faster way, understanding that pattern that I just described. And this is how we actually do it. Most amino acids, except proline, start with NH3. So you can do it, draw this peptide by saying, well, NH3 is connected to an alpha carbon, connected to C double bond O, connected to NH. And basically, at this position, you will write the side chain for valine. So I'm just going to write right here in the bottom, VAL, which is the three-letter code for valine. So I'm just going to color code this. So the valine side chain goes here. Once I have written the amide bond, which basically is this line right here, that's what this is representing. Then I'm going to write the next alpha carbon. So I draw alpha carbon, and then I will write double bond O, NH, which that double bond NH essentially is this one. Out of this alpha carbon, I'm going to draw acin. And remember that if you look at the table that specifically we have in the in-class worksheet, that is going to be the side chain for asparagine. So when it comes to this, at this alpha carbon, I'm going to write the side chain for asparagine. Continuing on, I need to draw an alpha carbon for cysteine. And notice that after cysteine, we still have a line, so we have to still draw a C double bond O NH. So at this alpha carbon, we're going to write the side chain of cysteine. And then you notice that after cysteine, there's the amide bond, which I already drew. But then after ASP, which is aspartic acid or aspartate, there is no line. So that means that that is the end of our peptide, which means that overall, I'm going to draw alpha carbon. And to conclude my peptide, I'm going to write C double bond O bonded to O negative. And essentially, I know to draw this at the end because there's no line as per aspartic acid. So the side chain for ASP is written right there. So to conclude my drawing, all I have to do is actually incorporate the side chains for the amino acids that I have here. So starting with valine. Valine side chain is an isopropyl group. Next, I have asparagine. The asparagine side chain includes a CH2 group followed by a C double bond O, NH2. Next, I have a cysteine. Cysteine is a CH2 connected to SH. And then, lastly, we have aspartic acid. And then for aspartic acid specifically, or aspartate, because remember that that means the same thing, aspartic acid or aspartate, then we have a CH2 connected to a C double bond O, And that's basically how we write a peptide. So far, I have mentioned the idea of a protein and an amino acid. And you have heard me use the word peptide. Let's formally actually define what is a peptide versus a protein versus an amino acid. A peptide is just going to be the units of amino acids coming together and making a polymer, which is going to be a molecule in which the amino acids are going to be the basic unit. And these amino acids are actually linked together via amide bonds. Now, a peptide or polypeptide is going to be an intermediate chain length containing up to 50 amino acids. Protein specifically is going to be an amino acid polymer that has more than 50 amino acids. An amino acid residue is just another word to talk about the amino acid subunit, that it is the basic unit found in peptides and polypeptides, and it's basically what we have in just proteins in general. It's just the basic unit. Remember that these amino acids are going to form peptide bonds, polypeptides, proteins, in other words, via the removal of water, as we just practiced when we were drawing peptides for a sequence of amino acids given. In figure 12.7, you actually observed an image of Frederick Sanger. Frederick Sanger was a biochemist that actually was awarded the Nobel Prize in 1958. And this scientist was awarded the Nobel Prize because he discovered the amino acid sequence for several proteins, but one of the main ones that people remember is the amino acid sequence for insulin. Insulin, it is a protein that contains 51 amino acids. Insulin, we're going to see later in this chapter that it is an important protein and it is utilized as treatment for people that have type 1 and type 2 diabetes, depending on the condition hallmarks. The next reaction of amino acids is going to be an oxidation reaction. Specifically, the side chain of a cysteine amino acid can undergo oxidation. When the thiol group of a cysteine amino acid encounters another side chain for a cysteine amino acid, the thiols, in other words, the sulfhydryl groups, the SH groups, via oxidation can form what is called a disulfide bond. This reaction is reversible. A disulfide bond can be reversed via reduction and gives back the 2SH group that we can see that is the side chain for cysteine. Interestingly, cysteine as an amino acid can sometimes crystallize in urine. And in figure 12.8, you actually see images of cysteine stones, which is just these rock-like structures that we can observe, or substances in a way, that come from crystallized cysteine. So you're just observing an image of it.